Hey there, physiology. It looks like an awful lot of the class is going to be showing up a little bit late. That's all right. We can get started anyway. Announcements. As usual, we have a weekly quiz and journal entry prompt that are due on Friday, February 24th. The next assignments will be posted on Friday, and those will be due March 3rd. The project is coming up quick. If you want to request your topic and what kind of format you're going to do the presentation in, those need to be in by Friday, March 3rd. If you get a 1 for your grade on this request, that means everything's approved, you're good to go, nothing to worry about. If you get a 0.5, that means I left you a message. Please go look for that note. It may be a good source of information. It may be suggestions on how to present the material or suggestions on either expanding or narrowing down a topic. And a 0 generally either means you're way off topic, like you want to talk about penguins or in epidemics class, or it means somebody's already gotten that topic and you need to try a different topic. Are there any questions about the project at this point? Okay. So we've been talking about smallpox, and more specifically about the first vaccine for smallpox. So the smallpox vaccine was originally created using cowpox in 1796. I remember somebody mentioned this fact. They said it was before germ theory, and you're absolutely right. Germ theory did not come about until 1861, and that was Louis Pasteur. And we're going to talk about how Pasteur figured out not so much germ theory, but the understanding of how vaccines work, which was even later than germ theory. It was 1879, and that was Pasteur and a gentleman called Chamberlain. So a well-known quote in, at least in science fields, is, "If I have seen further, it is by standing on the shoulders of giants." This is something I think that we need to be aware of when we're studying the history of science, because while Jenner created the first vaccine, he was building on work that had been done by people in other countries, who knows who knows when, where they used variolation to protect people. Germ theory and understanding vaccines, again, we're building on information that we have figured out in the past. You're not going to go from knowing nothing about diseases straight to germ theory and vaccines. There are steps that have to be made, and we tend to ignore those steps unless they're significant. So what we get is we get a bunch of people who are famous for taking information that others have developed and making an intuitive leap, going from a vague understanding to suddenly it clicks and they get it. The quote, "If I have seen further, it is by standing on the shoulders of giants," is usually attributed to Isaac Newton. But the concept, this not specifically these words in this order, but this idea, we don't know the origin of it. It was first seen in a text from the year 500, so well before Isaac Newton.
That seems kind of fitting then.
Yes, it does. Okay, so let's talk about how Pasteur and Chamberlain in 1879 figured out how vaccines work, or at least made some major progress on how vaccines work. And Pasteur does deserve credit for developing a whole bunch of vaccines for both human and animal diseases. Pasteur is on the left, Chamberlain is on the right, and there are some chickens in the middle. In 1879, Pasteur went on vacation. And Chamberlain was supposed to go on vacation too. And he was supposed to infect some chickens with a disease called avian cholera before he left for vacation. Avian cholera is a very fatal disease in chickens. It can cause something called septicemia, which is an infection in the bloodstream. And it generally has a case fatality rate of somewhere between 90 and 100%. So the idea was Chamberlain was going to infect the flock so that they could keep this culture, this captive disease going while they were on vacation. So they didn't have to come back and try and, find it again. They didn't have the fancy negative 80-degree freezers we have today, where we can store vials of viruses for long periods of time and have them still be viable or able to infect. So not surprisingly, Pasteur was the head of the lab. Chamberlain was working in the lab of Pasteur. So Chamberlain came back from vacation first, and he saw sitting out on the counter that vial of cholera he was supposed to infect the chickens with. Oh boy, come back from vacation and find out that you forgot something really important the boss told you to do. So he figured, well, better late than never. And he infected the chickens. And something weird happened. The chickens got sick, but they didn't die. Bizarre. It has a high fatality rate, but none of them died. So Chamberlain then made up a new batch of cholera. I'm not sure where he got the cholera from at this point, and infected the flock again. Again, some of the chickens got sick, none of them died. This is strange. So Pasteur returned, and Chamberlain had to face his boss. He had to go, oh God, I forgot to infect the chickens. I tried infecting them, but they didn't die. And I tried infecting them again, and they still didn't die. And I don't know exactly what's going on. And I'm so sorry, I screwed up. But Pasteur said, that's interesting. More scientific discoveries come from that phrase. That's interesting than from any other phrase. So he said, let's do the same thing again. And they got the same results. So what he realized was that if you leave cholera out on the bench and the cholera dies, it doesn't kill the chickens, but it protects them from live cholera. And so he basically figured out, this is how vaccines work. You show the immune system something that's not going to kill them, but you get them prepared to see something that could kill them. And famously, he said, chance favors the prepared mind. I didn't track down whether he was the first one to say it, probably not, but the concept was there, all because of an accident. So When we talk about vaccines, we have to understand, get a basic understanding of how the immune system works. The immune system is insanely complicated. We understand some of it, but definitely not all of it. This discussion is really skimming the surface of the immune system. It's really giving you a very simplified overview. People who go to graduate school will take whole semester-long courses just on the immune system. When we're talking about the immune system, there are two terms I want you to remember. Antigen and antibody. So antigen is a molecule, usually a protein, that triggers an immune response. And an antibody is a protein, usually, that attaches to a specific antigen as part of the immune response. Let me stretch this out so I'm not typing over words. There we go. All right. And of course, there's a cartoon over here on the left. This is XKCD, the science nerd's favorite cartoon. My body hosts an autonomous microscopic defense swarm that will do anything to protect me. I have no ability to restrain it, and I don't know my own power. So listen up. Sales grew by 4% this quarter. Business pro tip. You can strengthen any presentation by opening with a reminder of just how cool immune systems are. And now we're going to dive into it. So, when we talk about the immune system, we can divide it into a couple of different parts, different things that are involved. Things is a very vague word. But we're talking about cells and secreted molecules. So immune cells are generally produced in the bone marrow. They often have to go somewhere else to mature, like the thymus. The thymus is a organ right in your upper chest. And it's very important for maturing these immune cells and sifting through the mature immune cells to get rid of ones that could potentially be harmful to you and support the ones that are good for your defense system. Most cells that go to the thymus fail and are destroyed by the thymus. Then there are secreted molecules. A molecule is just, it can be a protein, it can be a carbohydrate, it can be a lipid. These terms are probably familiar to you. We talk about them when we talk about food, but they can also be small things like a water molecule or a molecule of carbon dioxide. So it's just a vague term for a bunch of atoms stuck together that's pretty small. We're talking about something small. And the secreted molecules that we're going to talk about are antibodies and histamine. There's a whole bunch of other ones we're just not even going to discuss. And our immune system targets include viruses, bacteria. That's largely what we talk about all semester, but it can also attack harmful fungi, prions, which are weird. They're just seriously weird. These are proteins that can cause neurological disease. So mad cow disease is one. Chronic wasting disease is one of these. They're just completely weird. Then there are protozoans. These are parasites. Well, I would put these two in the same category because generally parasites can be protozoans or they can be animals. But the protozoans that infect us generally act like parasites. Protozoan is a usually single-celled organism that are not animals, they're not plants, and they're not fungi or bacteria. The immune system targets cancer cells and foreign debris like splinters. So the function of the immune system is basically to determine if something is part of yourself or if it's foreign, if it's not part of the body, not part of yourself. And it protects you from things that are not yourself. This can include infected cells, cancer cells, infectious agents, but not healthy cells. So it does happen that the immune system protects healthy cells. That is called an autoimmune disease. And there's a whole bunch of these diseases. Crohn's disease, type 1 diabetes, some types of arthritis, lupus, irritable bowel syndrome. So there's a whole range of these, multiple sclerosis. And the autoimmune diseases can affect any part of the body. It just depends on which type of cell the immune system decides are abnormal and goes after them, even though they are healthy cells. So our basic immune response: detect and identify the pathogens, recruit other immune cells to the site of the injury or the location of the pathogens. So where is an infection or a potential infection? Coordinate the immune response, and then destroy or suppress the pathogen. So let's talk about our first lines of defense, and then we'll get into how the immune system, the innate immune system, the adaptive immune system, and how that all works. But the first line of defense is actually barriers. Barriers that prevent a virus, a parasite, a bacteria from getting into the body. And surprisingly, something that we don't tend to think about are the physical barriers. Your skin is the first line of defense against infection. If the bacteria cannot get through your skin, it can't infect you. And the surface layer of your skin is all dead. The cells that you see on the surface of your skin are dead skin cells. So you can't infect those. They're dead. So it's a really good first line of defense. We also have chemical barriers. Chemical barriers are things like stomach acid, the mucus in your nose. There are chemicals in your tears that help. These can do things like create a condition where there's an incredibly low pH. pH is a measure of acidity, and most organisms can only live within a certain pH range. So having a low pH makes it harder for bacteria or viruses to survive. That acidic environment can literally destroy these organisms or infectious particles. So while the skin and the vagina have relatively low pHs, the stomach really, really hits it. It has a pH of around 2, and that means that any food you swallow is going to be treated with acid first. And that's going to kill off the vast majority of potentially harmful microbes that get into your digestive tract. There are simple antimicrobial peptides. These would be produced by immune cells and secreted. And then a lot of mucous membranes, the membranes like inside our nose, inside our mouth, inside the anus, inside the urinary tract. These will secrete mucus. It's a molecule that's called mucin. And in the presence of water, it forms a thick, viscous substance that will make it hard for those microbes to get to the cells. It will help block access to the cells of the that line the inside of the body. It's like trying to swim in Jello. You just can't really do it very well. Some of these antimicrobial peptides include things like lysozyme. Lysozyme breaks down the cell wall of bacteria. Anytime you see this phrase LYS or LYZ, that's something that's going to break down or destroy something else. Another category of small molecules are defensins. These also disrupt bacteria cell membranes. So I've used two different terms here, and I know we haven't talked about bacteria yet. So let me try and give you an idea of what we're talking about. So all cells have a cell membrane. The cell membrane is primarily lipids, and it basically separates the inside of the cell from the outside world. We have these on all of our cells. Bacteria also have a second structure. Oops. Okay, why are you not letting me do this? There we go. Called a cell wall. That cell wall is a secondary structure outside the cell membrane that helps protect the bacteria from the environment. So we have two different types of antimicrobial peptides. Peptides are just tiny proteins. Lysozyme, which breaks down the cell wall, and defensins that go after the cell membrane. So they're going after two different parts of the bacteria. And these are both what we call broad spectrum, which means that they'll go after any bacteria. They don't try and go after, or they have the ability to go after a large diversity of bacteria. Not sometimes we'll see things that can only go after certain types of cell walls, because we have different types of cell walls in bacteria. Or it can only go after bacteria that have a certain protein on the surface. So these chemical barriers are just generalists. They'll go after anything. And then there's the microbiome. So on the surface of our body and lining the inside of all of our organs, the inside of the digestive tract, the inside of the lungs, the inside of the urinary tract, there are microflora. This is a fancy word that means there are bacteria and fungi and sometimes small animals like mites, microscopic mites, that are normal. They live on our skin, they live in our bodies. Many of them are beneficial. They can, in our digestive tract, they're important for digestion. If you sterilized the inside of your digestive tract, you would have severe malnutrition. We need these bacteria to digest certain foods, produce certain vitamins that we need. This is an intense area of research that we're really just beginning to understand. But one of the things that they do when we're talking about infection is they interfere with the ability of other microbes to attach to the surface, either the surface of the skin, the lining of the nose, or the mouth, or the digestive tract. They're like, we're already here. We're not going to let you attach. They're going to attack these microbes directly, and they consume any of our waste products that could provide a food source for the bacteria. So oils that we secrete from our glands, our microbiome consumes those so that other organisms can't. Okay, so that's our first line of defense, stopping an infection from getting started. How are you guys doing with this?
Good.
I'm good.
Yeah.
Okay, great. Now, when we have a situation where an infection has started, the second line of defense is what we call the innate immunity, or the innate immune system. Innate means inborn. We are born with this immunity. It's very nonspecific, so it can respond to a lot of different types of pathogens, including things we've never seen before. This is very important for new types of infections, things that we haven't seen. So when a new disease comes around, it can be something like a new virus that causes the common cold. The first time we see it, our innate immune system is going to go after it. And it has a relatively rapid response. So the idea is, this is something new, we're going to go after it and hopefully prevent it from becoming established and becoming a bigger problem for the body. There are several different mechanisms we're going to talk about. We're going to talk about something called phagocytosis, and I'll explain what that is on the next slide. They can release chemicals and proteins to help fight off bacteria, things like the defensins that I mentioned, lysozyme. And then they trigger physiological processes that help, things like inflammation and fever. And I just put this picture over here to give you the general idea that not all the immune system has lots of different kinds of cells. We're going to lump them all together in the category white blood cell, but the different types of cells have different specific functions. We'll talk about a few of them, but we're really not going to cover very many at all. Okay, phagocytosis. This is a big, probably unfamiliar word. So let's look at this word. Phago means eat. Cyte means cell. So phagocytosis is a process where a cell eats something. Scientific words often can, if you start learning these little segments and what they mean, they can really help you understand unfamiliar words. So you can start learning how to break down these words so that you can figure out what they mean without having to just learn from scratch. Okay, so phagocytosis eats the pathogen. It starts hang on, looking at my notes. I've changed my notes. So the cells that do this, they have these receptors on the surface. Receptors is right here. These are what are called pattern recognition receptors, and they recognize specific molecular patterns that are often associated with pathogens. So some things that are common on the cells of bacteria or common on the cells of viruses, these cells that are capable of phagocytosis recognize that. They also recognize what's called damage-associated molecular patterns. These damage-associated molecular patterns are often cellular debris. So there'll be things that should be inside of a cell, but if a pathogen comes along and kills a cell, the cell will break down, and these molecules that should have been kept inside the cell will now be out. And they'll recognize those and eat those as well. These attacks are not always successful because a lot of, especially bacteria and some viruses, have learned how to hide from these types of cells. These types of cells are called phagocytes. Phago-eating cyte cell. One other thing that these guys can do, which is really cool, is they can take a molecule from a pathogen and present it to other cells of the immune system. This is called antigen presentation. We're going to talk about that more when we talk about the adaptive immune system, because it's very, very important in the adaptive immune system. All right. So those are, that's phagocytosis, which is one process. Then we have these other cells in the white blood cells called leukocytes. And these are important with mediating the immune response. Specifically, it is trying to reduce inflammation, reduce allergic reactions. So they both, this category of cells works both to target specifically larger parasites. So it makes these cells called, or make these molecules called cytotoxins. Cyto, cell, toxin, and that's just what it says, it's a toxin. So it produces these molecules that target parasites specifically. These cells, as I mentioned, try and moderate allergy, but if they don't do their job correctly, they'll often be in, when they don't do their job correctly, you often end up with allergies and asthma. So an allergy is your immune system overreacting to something, usually something that is harmless to the body, like tree pollen, peanuts, cat fur, all of these sorts of things. Our body responds to them. They're not going to kill us, but our body decides that they're the bad guys and overreacts because the leukocytes are not working correctly. There's one other thing that the leukocytes are important for, and that is a process called apoptosis. So this is probably an unfamiliar word that you haven't heard before. And apoptosis is a way of breaking down a cell that is damaged or infected, that recycles material and prevents release of potentially harmful substances. So we talked about lysozyme, which is something that breaks things down. Well, all of our cells have a special miniature organ, an organelle, that has lysozyme in it. We don't want that getting out uncontrolled. So if we have an infection that's killing a lot of cells, if we can have them do apoptosis, we can prevent the release of that lysozyme into the area where it can cause damage to uninfected cells, basically. So this slide is all about inflammation. It's probably a word you've heard before. It's commonly talked about by doctors and medical experts. It seems to be involved in chronic inflammation, seems to be involved in some aging processes, some types of things like heart disease. I say probably, that's not my field of study and I'm not up to date on that. But inflammation is a localized immune event. And it is designed to prevent spread of the pathogen. Generally speaking, inflammation is characterized by heat, pain, redness, and swelling. So we've all had times where we got a cut or a splinter and it got kind of red and painful to the touch and a little bit swollen. All of that is to try and prevent the pathogen from spreading and to take it out before it can spread. So there's a couple of different molecules that are released, one of which is heparin. You've probably heard of heparin. Heparin it prevents clotting. It prevents the formation of clots. We want to do this because we want white blood cells to be able to get to the site of the infection. And if the pathogen damages the blood vessels, it can cause clots. Clots are a natural response to damage by blood vessels. So heparin stops that so we can bring the white blood cells in to attack the potential pathogen. It also produces chemicals that cause vasodilation. This is enlarging the capillaries, the very, very small blood vessels that basically go everywhere. It opens those up. Again, we're increasing the ability of other white blood cells to arrive. It increases the permeability of capillaries. So what this means is that these white blood cells that are now being attracted to this area have an easier time of getting out of the capillaries and into the surrounding tissues where the infection is. So a lot of the immune response is to bring white blood cells to the area. The innate immune system produces chemicals that attract cells that are capable of phagocytosis, for example, as well as some other white blood cells. And then it also stimulates pain receptors. So we said there's heat. Heat comes from two things. One is increased blood flow, and the other is increased metabolic activity. When your cells are extremely energetic and they're doing a lot of different things, heat is a byproduct of that. It's a side effect. So heat is a sign that the cells are very, very active. Pain is directly because of chemical irritants released by some of the innate immune system. This is telling you you got injured. We need to know when we're injured. Otherwise, we would do the same stupid things over and over again and not realize that we're getting injured. People that don't have a good pain response tend to have lots more injuries than people who have a typical pain response. They tend to have lots more scarring. They're more likely to have serious injuries because they're not aware of the injury happening. We all think of pain as a bad thing, but a lack of pain is also a bad thing. Redness, increased blood flow, and some leakage from the capillaries where we have that increased permeability. And then swelling. Swelling is caused again by that increased permeability. There's fluid that comes out of the capillaries into the area. This fluid will eventually collect the pathogens, collect the cellular debris, and carry it into the lymphatic system. So the lymphatic system is something that we know about lymph nodes usually. We can feel them sometimes under the line of the jaw, the back of the neck, the armpits. Lymph nodes are a location where the body stores white blood cells. They have a net-like structure inside so that they catch bacteria, cancer cells and cellular debris, for those immune cells that stay in the lymph node to finish breaking down, to completely destroy those. But there's also a series of lymph vessels. It's similar to the circulatory system where we have blood vessels everywhere, but there's no pump involved. And these move liquid around the body. Some of it is to make sure that liquid is distributed appropriately around the body, and some of it is to filter that liquid through the lymph nodes to get rid of that cellular debris and bacteria. So that's why we have inflammation. You guys keeping up with me?
Yep.
I think so.
Think so. Okay. It's complicated stuff, and I am really simplifying it for you guys. So let me know if you need me to explain something again. You can either interrupt me, raise your hand, or put a question in the meeting chat. I've got the chat box open. Okay. Oh, here's another slide on inflammation. So again, this is basically what I talked about last time. We've got bacteria. This looks like a splinter that came in through the skin. This is the skin here. And we have cells from the bloodstream slipping out through that permeability and attacking the bacteria. Fever. Part of your innate immune response, your response to unfamiliar organisms, is fever. This is a temporary increase in body temperature due to an infection. Generally, in adults, fever is over 100.4 Fahrenheit. And it's dangerous over 103 or 104. In young children, higher fevers are not necessarily as dangerous as they are in adults. So those numbers are for adults. Why do we want to raise the body temperature? We're doing this in part to increase cellular activity. Our cells, when they're more active, produce more heat. More heat makes them more active. So it's kind of a reinforcing cycle. This increased rate of cellular activity includes the immune response and cellular repair mechanisms. The immune response and the pathogen both cause damage to cells. So the cells have to be able to repair themselves, if possible. It causes stress to pathogens. It causes stress to infected cells too, but it really causes stress, especially to bacterial pathogens. It makes it harder for them to replicate. So bacteria multiply by each cell gets larger and they physically separate. So they double very, very quickly. And that is reduced, that ability is reduced by the high fever. In general, a high fever is just something you should kind of just ride through. If it gets too high or it goes on too long, you can take fever reducers. And a lot of doctors will recommend that just because you'll feel better. You know, it's maybe sometimes it's helpful to have a fever, but sometimes it's not. And, you know, so you get conflicting advice on that one. So how high should the fever be before you go to a doctor? For most ages, it's 102. Although if a fever is continuous for several days, you should also see a doctor. Hang on, I'm having trouble with mine. However, there are a couple of very important things. Do not give aspirin to children when they have a fever. Adults can take it, but children should get either ibuprofen, Tylenol, Advil, some other form of fever reducer. Adults should take a fever reducer if the fever is over 101. If it's lower than that, you know, you might want to let your immune system try and do its job there. And over 103 or staying high after treatment, seek medical care. Actually, I said 102 earlier, so let's stick with 102. Again, very young children, a temperature of 103, 104, call the pediatrician and ask for advice, because they may just tell you to give them fever reducers. So that's the innate immune response. That is the first, the way the body responds first to an infection, especially a new infection. It's kind of hard on the body. It's very fast, but it's not very strong. So we have a third line of defense, which is the adaptive or acquired immune response. This is a slower initial response. It takes longer to get going, but it is specific to an individual pathogen, and it is a much stronger response. In addition, the adaptive immune system has the ability to remember. So it can remember a pathogen. This is going to be important for vaccinations. So when we look at the adaptive immune system, all white blood cells start out from the bone marrow, especially the long bones, like the upper leg, the femur bones. With the adaptive immune response, we have B cells and T cells. T cells are cells that have gone to the thymus. B cells are straight from the bone marrow. And they mature into a range of different cells. And the B cells will eventually produce antibodies. The T cells, we'll talk about exactly what they do later. One of the advantages to this system is that these cells, these T cells, T lymphocytes, and B cells or B lymphocytes can multiply very quickly and produce huge numbers of cells to tackle an infection. This is not something the innate immune response can do very well. And so when we look at these two types of cells, the T lymphocytes do something called cell-mediated immunity. So the cells are going to be in charge of managing the immune response and actually doing the immune response. B lymphocytes can do cell-mediated immunity as well. And we're going to talk about that in just a second. But they also produce antibodies. Antibodies are a powerful tool in our immune system. They do a lot of the work in our immune system. Very important. Okay. Certain of these white blood cells have the ability to do antigen presentation. And I've given you a list here, but I'm not expecting you to know these, except for the B cells, the bone marrow cells. There are some other cells that can do this. Regardless, these cells will phagocytose the pathogen. They'll eat the pathogen. They'll take the antigen from the pathogen, something that's distinct to the pathogen, and present that to a T cell. The T cell will then produce a response. Okay. This immune system part is probably the hardest part of the whole semester. So try and stick with me, ask me questions, interrupt me, put something in the chat if you need me to go over it again, or watch the video once I post it on YouTube. Okay, T cells. These are the ones that go to the thymus to mature. Mature T cells can bind to or recognize the antigen presenting cells and learn to recognize that antigen. And we have three major categories of T cells. There are cytotoxic T cells. We've used this word before. Cyto means cell, toxic means, well, toxic. These go after virus-infected cells, causing them to do apoptosis, which is, remember, that's a controlled way to destroy or kill the cell so that everything can be recycled. We're not just getting the virus released in large quantities. It's a controlled process. Cytotoxic T cells also destroy tumor cells. These are cancer cells or cells that could become cancerous, again, using apoptosis. The helper T cells secrete chemicals to increase the immune response. These include the category of CD4 positive cells. Remember what CD4, we've talked about CD4 positive cells before. When did we talk about it? Does anybody remember? No? All right.
I want to say we talked about it when we were talking about HIV.
Exactly.
Yeah.
That's exactly right. These are the cells that HIV infects. So these chemicals help to coordinate the innate immune response, the adaptive immune response, and what we call the humoral response, which is basically mostly antibodies. And then there are regulatory T cells. These suppress the immune response. These are the off switch to the immune response. So when we have an immune response, we need to keep it balanced so that it's strong enough to destroy the pathogen, but not strong enough that it becomes an autoimmune disease or causes runaway inflammation. You can have an immune response that is so strong that the immune response ends up being damaging to the body or even fatal. A lot of the early COVID cases, we were seeing this, where the immune response created a cytokine storm, is what they were calling it in the news. I'm not going to ask you what a cytokine storm is because we're not going to talk about cytokines, but it was an overactive response from the immune system. So we need regulation and control. The B lymphocytes, this is what we call the humoral branch of the adaptive immune response. These are activated by helper T cells. There are a few other things that can stimulate this. These B cells are produced in the bone marrow. That's where the B comes from. And the most important thing about these is they produce antibodies. So when they are activated by a helper T cell, they differentiate into change, become different into larger cells that produce huge numbers of antibodies against a specific antigen. Antibodies are very specific. They have this distinctive, almost Y shape, and they will attach to the antigen. That antigen can be on the pathogen, it can be on the surface of an infected cell, and this tag is a signal to other cells of the immune system saying, come after this, come get this. They can also block activity of a pathogen. So if the cell surface molecules of the pathogen are covered with antibodies against specific antigens on the surface of that pathogen, that can physically block the ability of that pathogen to attach to a target cell and to get into that target cell. So it's a physical block. Each of these cells, these B lymphocytes and T lymphocytes, have receptors, molecules on the surface that are specific to a distinct antigen. So they have a molecule on the surface that is designed to recognize a specific antigen from a specific pathogen. When that antigen binds, I mentioned this in the last slide, they become activated. And this can be from a T cell presenting the antigen to it, or just bumping into that bacteria or virus or infected cell. And they multiply like crazy. They produce large numbers of identical cells that all recognize that antigen. This takes a long period of time, relatively speaking, but it causes a very strong immune response. So we end up with very large numbers of cells that are capable of targeting a specific pathogen. When I say it takes a long time, we're talking 1 to 2 weeks. So let me put that down here, 1 to 2 weeks. The time between exposure to this kind of response is lag time. Now, how can we have enough lymphocytes to recognize all of the potential, all of the bacteria and viruses and everything that we've been exposed to? You have millions of different B lymphocytes in your body. We just have tons of these things. We make lots of different antigens on the surface to be able to go after lots of different potential things that are harmful. We also remember, we're going to get to the memory cells in the next slide, but we also have the ability to remember a huge number of things that we've been exposed to. And so now we're getting to memory cells. So this graph on the left, the x-axis is time in days. Notice there aren't any hash marks. This y-axis is how many antibodies are in the bloodstream. So here is your first exposure to an antigen. We have our innate immune response, which is kind of weak, doesn't produce antibodies. After 1 to 2 weeks, we get our primary adaptive immune response, and we get antibodies produced. So this is 1 to 2 weeks after the first exposure. The number of antibodies will drop, but not go back down to 0. And then at some point in the future, if we see the same antigen, we have long-lived B and T cells that remember that antigen and are producing a low level of antibodies at all times. Having some antibodies in the system and these cells that remember those antigens means the adaptive or acquired immune response can kick in faster the second time you see a specific antigen. So we end up with a much faster and stronger immune response. This is only going to take a day or two instead of the week or two that it took the first time. And what this means is your immune system's going to be able to go after the pathogen before it gets established. It's going to have a strong response. You're going to have a milder illness if you have an illness at all. So you may have no symptoms of infection. Okay. That is the immune system in one mind-blowing lecture. And that is the hardest lecture you're going to have in this class this semester. So look over the notes, watch the video again if you have to, email me questions if you need to, or you can ask me questions now. But I'm going to go ahead and stop us here because I don't want to get started on how vaccines work. But vaccines are basically going to be acting in place of your first exposure to an antigen. So they're going to be exposing you to something that is not capable of causing you to be sick so that you have a faster response if you do get exposed to that same antigen. Okay. Any questions?
Sounds good.
Okay.
Thank you. Have a nice one.
Thank
Okay.
you.
See
Bye.
you
Thank
guys
you.
next time.
Have a nice day.
Bye.
Thank you.
Thank you.
Bye.